In the late 1980s, microbiologists kept reading bacterial DNA for practical reasons, to map genes, to track traits, to understand how microbes worked. But every so often, the sequence printouts delivered something that did not fit the assignment. A short stretch of DNA repeated again and again, with the repeats spaced at regular intervals. It looked orderly, almost engineered. It also looked pointless. These repeats did not behave like known genes. They did not code for proteins. They did not resemble the common mobile elements scientists already blamed for odd patterns. And they sat in places where researchers expected useful information, yet they gave none. In paper after paper, the repeats appeared as a brief note, a curiosity in the margins. The work moved on because no one had a reason to stop. Still, the patterns stayed hard to ignore. The repeats were nearly identical, and the gaps between them were not random lengths. Something kept placing them there, or maintaining them with surprising consistency. But at the time, sequencing was slow. Databases were thin, and comparisons across species were limited, so the repeats remained a question without a handle. They were recorded, archived, and left unexplained. Through the 1990s, DNA sequencing spread from a handful of labs into a growing engine of discovery. Each new bacterial genome arrived like a fresh map, and the maps started showing the same odd landmark. The repeating DNA pattern kept turning up again, not once, not twice, across species that lived in different places and behaved in different ways. That repetition mattered, because biology rarely kept useless details identical for long. As researchers compared genomes, the pattern looked strikingly consistent. Short repeats spaced out in a regular rhythm, with unique bits in between. Different microbes carried different versions, but the overall layout stayed recognizable. It appeared in bacteria, and it appeared in archaea too, a whole other branch of microbial life. That reach changed the question. It no longer sounded like a sequencing accident. It sounded like an old solution, copied and preserved. By the end of the decade, the evidence piled up in plain sight. The repeating arrays showed up so often that ignoring them became harder than noticing them. If so many unrelated microbes carried the same kind of DNA structure, it probably did something important, yet no one could say what that function was. The field sat in a frustrating middle ground, a pattern that looked conserved on purpose, but still had no clear job. By the early 2000s, the repeating DNA pattern stopped looking like a lonely oddity and started looking like it belonged to something larger. As more complete genomes arrived, researchers noticed a clue that kept appearing right next to the repeats, a cluster of other genes, positioned like a package deal. These genes looked conserved, organized, and intentional. The repeats and these neighbors traveled together. That pairing suggested purpose. Those neighboring genes became the turning point. They encoded proteins, and they appeared so often beside the repeats that the link felt hard to ignore. Scientists began to treat the whole region as one unit, not two separate observations. The repeats provided a recognizable signature, but the nearby genes hinted at machinery. A pattern became a system. Suddenly, there was something testable, something that could act. With that shift came a need for order. Different labs had used different labels, and it slowed everything down. A standardized name emerged: CRISPR, short for clustered, regularly interspaced, short palindromic repeats, and the neighboring genes received a matching label, CRISPR-associated, or Cas. That naming did more than tidy up vocabulary. It gave the field a common target, and research moved from noticing to investigating. With CRISPR now recognized as a real system, attention shifted to the most mysterious pieces, the spacers, the short DNA segments sitting between the repeats. At first, they looked random, different from one microbe to the next, with no obvious purpose. But genome databases grew, and researchers started comparing spacer sequences to known genetic material. That simple comparison changed everything. Some spacers looked strangely familiar. Again and again, spacers matched fragments of viruses that infected bacteria, and also plasmids, small DNA circles that jumped between microbes. The matches were not vague. They lined up like copied text. That meant the spacers did not come from the microbe's own genome. They came from outsiders. Each match added pressure to a new idea. CRISPR might not be a passive pattern at all. The implication was sharp. If spacers came from invaders, then the CRISPR array looked like a timeline, bits of past attacks stored in the DNA, one after another. Not a diary written in words, but a genetic scrapbook of infections and encounters. That reframed the entire field. Instead of asking what CRISPR was made of, scientists asked what it remembered and what that memory might be used for. The big question stayed simple. Did those spacer sequences just sit there like fossils, or did they actually do something? Researchers tested it the most direct way they could. They exposed microbes to a virus, watched which cells survived, and then checked the CRISPR region. A pattern emerged. Survivors often carried a new spacer that matched the very virus that had attacked them. That detail changed everything, because it meant the system did not only remember the past, it learned from the present. The microbes had captured a piece of the invader's genetic code and stored it in their own DNA. And when scientists challenged those same microbes again with the same virus, the outcome shifted. Cells with the matching spacer resisted infection far better than cells without it. Then the experiments tightened the link even more. When the spacer matched, protection followed. When the match failed, the virus broke through. And when researchers removed or disrupted the CRISPR system, the defense collapsed. Cause and effect lined up. CRISPR acted like an adaptive immune system for microbes, built from stolen fragments of past attackers and used as a real-time shield against reinfection. Once CRISPR clearly acted like immunity, the next question stayed brutal and simple. How did it actually stop an attack? Step by step, the parts lined up into a working chain. The CRISPR DNA got copied into RNA. Those long RNAs got chopped into smaller pieces, each one carrying the sequence of a past invader. And those small RNAs did not float alone. They locked onto Cas proteins and turned them into guided weapons. With that RNA guide attached, the Cas complex started behaving like a search tool. It moved along DNA and checked letters against the guide, almost like matching a fingerprint. When the match fit, Cas acted as a cutter. The foreign DNA broke, and a virus that needed intact genetic material could not keep going. The mystery shifted from whether CRISPR worked to how it avoided the worst mistake of all. Why did it not cut its own CRISPR memory? The answer came from a tiny rule sitting next to the target. The system required a short extra DNA tag beside the matching sequence, later called a PAM. Invader DNA often carried the PAM next to the target, so cutting proceeded. But inside the bacterium's own CRISPR array, the same spacer sequence sat without the right PAM beside it. No PAM meant no attack. It was a simple checkpoint that let CRISPR recognize the enemy without turning on itself. In 2012, the question changed from what CRISPR did in bacteria to what it could do anywhere. Researchers focused on a system called Cas9, a protein that cut DNA, but only when it was steered by RNA. In nature, that steering came from multiple RNA pieces. In the lab, scientists started rebuilding the parts from scratch, aiming for something simpler and controllable. they discovered they could merge the natural RNA components into a single guide RNA that carried two jobs at once. It latched onto Cas9, and it carried a short sequence that matched a DNA target. Change that short sequence, and Cas9 went to a new address. Nothing else about the protein needed to change. The system became programmable, like swapping a tip on the same tool. When the guide RNA matched, Cas9 cut the chosen DNA at that spot, reliably, on command. That was the breakthrough. A general method for making precise cuts in DNA without redesigning a new protein every time. With one Cas9 and a custom guide, researchers could target almost any sequence they selected. CRISPR stopped being only a microbial defense trick and became an engineerable genome editing method. In 2013, the big question was simple. Could CRISPR-Cas9 work inside the more complicated cells of plants and animals? Bacterial tricks often failed there. Eukaryotic DNA sat packed into chromosomes, guarded and managed in ways bacteria never dealt with. But when researchers delivered Cas9 and a designed guide RNA into these cells, the cut happened where it was supposed to. It was not a one-off. It was repeatable. That single shift changed the pace of biology. Once the cut could be made, cells tried to repair it, and that repair created edits, knockouts to disable a gene, or targeted changes when extra DNA templates were supplied. Labs that had never touched bacterial immunology started using CRISPR as a standard tool. Weeks replaced months. Experiments that used to demand specialized teams became routine steps in a workflow. The effects spread fast. Plant scientists edited crop traits and tested gene functions without years of breeding. Animal researchers built disease models by changing single genes and seeing the consequences in living organisms. In biomedicine, CRISPR turned genetics into something you could actively test, not just observe. It accelerated drug target discovery and made old questions newly practical. One tool moved from clever to unstoppable. The moment CRISPR started working in many kinds of cells, it stopped being just a scientific tool and became valuable property. Universities and research teams filed patents fast, each claiming they got there first, or got there in the most useful way. The fight turned public and expensive. Courts, filings, and licensing deals began shaping who could use CRISPR, how much it cost, and which companies moved first. These patent decisions did more than reward inventors. They steered the whole market. Some labs and startups gained broad access through licenses, while others faced uncertainty or higher fees. Investors watched the legal outcomes as closely as the lab results. Competing patent claims also created a split reality. CRISPR looked simple in principle, but building a product required navigating permissions, overlapping rights, and the risk of being blocked later. Then the most sensitive question arrived: editing human embryos. The technology made it possible to change DNA at the very start of life, meaning the edits could be passed to future generations. That possibility triggered worldwide alarm. Scientists and governments argued over limits, safety, and consent that could never be obtained from the people most affected. The controversy forced a new debate, not just what CRISPR could do, but what it should do. By the time the dust settled from the early rush, CRISPR no longer behaved like a single invention. It worked more like a toolbox. Researchers used different Cas proteins for different jobs, choosing the one that fit the target and the setting. The goal shifted from proving it worked to making it work safely, predictably, and in real patients. That change mattered. It turned a lab trick into a medical strategy. Clinical therapies then moved from promise to practice. Teams targeted diseases where a precise genetic change could bring a clear benefit, and where doctors could track results over time. Delivery became the make-or-break challenge. Editors had to reach the right cells at the right dose without causing dangerous cuts elsewhere. Each trial forced the same hard question. Could gene editing deliver real health gains without trading them for new risks? Next-generation editors pushed the idea even further. Some approaches changed single DNA letters without making a full break, while others rewrote short stretches of code with more control. As the tools diversified, the rules around them had to evolve too. Regulators weighed speed against caution, and policies shifted as new data arrived. The contrast stayed striking. What began as a weird repeating pattern in bacteria ended as a platform that society had to govern.